Good afternoon, everybody. Thanks for coming to today's Dean's Lecture. I'm Mike Clegg, and I think many of the people in the room know that these lectures are held when we appoint or promote somebody to the rank of professor. And you know, it's not easy to get on the faculty at Johns Hopkins. But it's an incredible task to rise to the level of professor. And as I often say, the decision is not made by me or the A&P committee. It's really made by peers all around the world who we ask for evaluations of the candidate. And so it's, I often feel like I'm cynical and jaded, but it's actually a very big deal to be promoted to professor. And so we're here today to celebrate and congratulate Liz Selvin for being promoted to professor. She's internationally recognized in the field of diabetes and cardiovascular disease epidemiology. Her work on hemoglobin A1c, which I'm sure you're going to hear about, we'll hear about hemoglobin A1c. So, and its association with complications and its role in the diagnosis of diabetes has really changed clinical practice and as well the field of diabetes epidemiology. She's got a special interest in the intersection of her science and public health and clinical policy. And much of her work focuses on biomarkers and diagnostics relevant to diabetes and complications. She's currently PI on grants from AHA, the American Heart Association, the National Institutes of Health, and the National Kidney Foundation. And in 2013, she was awarded the Harry Keene Memorial Award by the International Diabetes Federation for her contributions to the field of diabetes epidemiology. She went to Berkeley High, received her BA in political science from Northwestern, summa cum laude, and received Phi Beta Kappa and departmental honors. She made an error in going to the University of Michigan for her MPH, but has recovered adroitly and came here for her PhD, which she received in 2004. She was a fellow on the NIH-NHLBI cardiovascular disease epidemiology grant and was promoted to professor last year. She holds a joint appointment in the School of Medicine in the Division of General Internal Medicine and received the Advisory, Mentoring, and Teaching Award from the Student Assembly in our school in 2015. So please join me in welcoming Liz Selvin. Professor Selvin.
Thank you all for coming. It's great to have so many colleagues and family and friends here today. I think hopefully the students understand they're not actually required to attend. This is not my usual 8:30 a.m. lecture. So what is diabetes, to start at the beginning? So this is a very simplified diagram illustrating normal glucose homeostasis. Typically, blood glucose is fairly tightly regulated between 70 and 150 milligrams per deciliter in the blood. Insulin is the principal hormone that regulates glucose, secreted by the beta cells in the pancreas and acts on the liver and other tissues to suppress endogenous glucose production and stimulate glucose utilization. So in type 2 diabetes, there's a number of pathophysiologic abnormalities that act to raise glucose in the blood. So first we have resistance of the cells to the effects of insulin. We get failure of the beta cells to produce adequate amounts of insulin and excessive hepatic glucose production. So all of this to say is that individuals with type 2 diabetes are chronically exposed to elevated glucose levels in their blood. Diabetes is associated with a number of very serious complications. The vast majority of deaths in people with type 2 diabetes are from heart disease and stroke. Hypertension is common in people with diabetes. Retinopathy is now the leading cause of blindness in the United States. Kidney disease, a very important complication of diabetes, and diabetes is now the leading cause of end-stage renal disease in the US. Diabetes causes damage to the nervous system. It's also a leading cause of amputation. Periodontal disease can cause serious problems in pregnancy and a number of other complications. So diabetes is really associated with very serious health consequences. So we can't talk about diabetes without talking about the epidemic of obesity. So to me, this is sorry, I don't have my usual Somer Hall equipment. So to me, this is a really, really staggering figure. So this shows the prevalence of obesity or an elevated BMI. And so we see in the 1960s that only about 13% of the US population was obese. And moving forward here to the most recent data we have available from the National Health and Nutrition Examination Survey, we can see that almost 40% of the US population is obese. So an absolutely huge increase in prevalence of obesity in the United States. So obesity is really the main driver of the diabetes epidemic. And this sort of quantifies this really tight link between obesity and diabetes. So these are the relative risks of diabetes, so the development of diabetes by BMI in women and men. And so these are the relative risk ratios, so the risk at high BMI levels compared to a normal BMI. And so you see this incredibly steep and linear risk gradient between high BMI levels and subsequent risk of the development of diabetes. And for some perspective on this, right, we think of a strong association between an exposure and an outcome as smoking and lung cancer, for example. Right? So the relative risk of smoking and lung cancer, those estimates are usually around 20, 30, 40. So we're seeing relative risks that are as strong as the association between smoking and lung cancer, and in some instances even higher. So an incredibly strong association between obesity, body mass index, adiposity, and the subsequent development of diabetes. So these are the trends in diagnosed diabetes in the US, and these look a lot like the trends that we see with obesity. So in the 1960s, very few individuals in the United States with a diagnosis of diabetes, and then we've seen this very steady increase and a very rapid rise in the last decade or so. And the most recent projections suggest that things are not likely to get better anytime sooner. This is not just a US phenomenon. I don't need to tell folks in this room. This is happening all over the globe. Diabetes and obesity are a global epidemic and two of the most important public health challenges that we face currently. So some facts. So diabetes is a global epidemic. It's largely driven by the epidemic of obesity. I think that epidemiologic research is critical to informing a clinical and public health policy related to diabetes. And I'm going to try to demonstrate today that I think that rigorous epidemiology can help us separate out some of the facts from fictions or myths in the field of diabetes. So I think everybody knows a myth is a widely held but false belief or idea. And so there's this television show where these guys go out and they use science to, see if these myths can stand the test, of the scientific method. So that's sort of loosely the approach that I've taken in my career. My partner in crime in most of this research is Joe Koresh. And you can also see my incredible skills with Photoshop here. Okay, so to begin with, I'm going to talk about 4 different popular beliefs that I've worked on and tried to bring evidence to bear related to diabetes. So the first popular belief, one of the ones that I encountered first in my career, was this idea that glucose is best for diagnosis of diabetes. That hemoglobin A1c should not be used for diagnosis. It's only useful for monitoring glycemic control. So I'm going to show you evidence today and work that we've done to show that hemoglobin A1c is actually a very powerful, very helpful diagnostic test and actually has a number of advantages over glucose. Okay, so for those of you who don't spend all your time thinking about biomarkers of hyperglycemia, these are the 3 standard tests that we think about when we think about measuring elevated glucose levels or hyperglycemia. Fasting glucose, which is glucose measured at a single time point. Postprandial or 2-hour glucose, which is glucose measured after ingestion of a carbohydrate challenge, and then hemoglobin A1c, which is an indirect measure of hyperglycemia. And I think, as Mike alluded to, everyone knows is my favorite molecule. And so those of you who don't spend all your time thinking about the epidemiology of hemoglobin A1c, just some brief background, which is that glucose binds to hemoglobin in your red blood cells. And because your red blood cells turn over approximately every 120 days, you can measure the proportion of hemoglobin that's glycated. And that gives you a measure of average glucose exposure in the previous 2 to 3 months in an individual. The first assays for hemoglobin A1c became available in the 1970s. And so since then, it's been used as a standard measure to monitor glycemic control in people with a diagnosis of diabetes. So it's, for many decades, been this standard measure to guide treatment and medical decision-making in people with diabetes. So, and a lot of this evidence base for the use of hemoglobin A1c for monitoring and guiding our treatment decisions is based on a large literature establishing strong associations between hemoglobin A1c and small vessel disease, particularly retinopathy. And we know from evidence from randomized clinical trials that lowering hemoglobin A1c can prevent or slow the progression of the development of microvascular disease in people with diabetes. So there's a number of advantages of hemoglobin A1c for diagnosis of diabetes. So as this integrated measure of average glucose exposure over time, it has much less biological variability, especially compared to fasting or 2-hour glucose. So my hemoglobin A1c today is likely to be identical or very, very similar to my hemoglobin A1c measured a few weeks later. So it has this nice stable properties. It's a better index of overall glycemic exposure compared to glucose or 2-hour glucose by its very nature. As an assay, it's now very well standardized. We've maintained calibration across all laboratories. It also eliminates the need for the patient to be fasting, right? You don't need time samples. There's no need or preparation for a clinician visit. It can be measured at any time of the day, regardless of fasting status. And relatedly, it's relatively unaffected by acute factors. So recent physical activity, diet, et cetera, don't affect hemoglobin A1c. And we've used hemoglobin A1c for decades as a main metric for guiding and adjusting treatment in clinical practice. So some of the issues related to thinking about tests and diagnosis in diabetes is that type 2 diabetes has this gradual onset. Glucose levels rise over time, and our tests for defining diabetes are imperfect. So this actually really opens the doors or creates this gap where epidemiologic data can be very helpful to inform how glycemic biomarkers relate to health outcomes. So I showed you there's this very strong established link between hemoglobin A1c and retinopathy. But I also showed you that the leading cause of death in people with diabetes is cardiovascular disease. Right? The vast majority of deaths and morbidity and mortality in people with diabetes are from cardiovascular outcomes. At the time that we started this research, there was very little data on hemoglobin A1c and its relationship to long-term outcomes like cardiovascular disease in an initially non-diabetic population. So this was work that we did. I'm very fortunate to be an investigator of the atherosclerosis risk and community study. So one of the first things we did is we demonstrated that you could measure hemoglobin A1c in stored whole blood samples. So this was work done with Mike Steffes at the University of Minnesota laboratory. And so once we had demonstrated that you could validly and reliably measure hemoglobin A1c in samples that had been stored for over a decade, we subsequently measured hemoglobin A1c in over 14,000 participants in the ERIC cohort. And so then we could relate hemoglobin A1c values in an initially non-diabetic population to the subsequent development of diabetes, to coronary heart disease, stroke, and all-cause mortality. And this work was done in collaboration with Fred Brancati and Joe Koresh, my myth-busting partner in crime here. And what we demonstrated in this study was that hemoglobin A1c levels at levels greater than 6%, those individuals are at high risk for not only the development of diabetes, but also for coronary heart disease, stroke, and all-cause mortality. We also demonstrated that hemoglobin A1c is superior to fasting glucose for the assessment of long-term prognosis in this initially non-diabetic population. And this really helped establish this strong link between hemoglobin A1c and future vascular risk, and provided supporting evidence for the use of hemoglobin A1c as a diagnostic test. So these data were very helpful in informing clinical practice guidelines. And in 2010, the American Diabetes Association first recommended the use of hemoglobin A1c for diagnosis of diabetes. So this was a major change to clinical practice guidelines. So this is in 2010, this is the first time that hemoglobin A1c appeared here in this table for the under the criteria for diagnosis of diabetes. So it was added to glucose and 2-hour glucose as a biomarker that could be used for diagnosis of diabetes. You'll note here that these are numbered. A1c is listed here at the top. And so subsequently, hemoglobin A1c has now been formally incorporated into guidelines adopted by the WHO, the IDF, other international organizations, and we've now been using hemoglobin A1c for a diagnosis of diabetes for the last 6 years. So I think you can see that hemoglobin A1c is a useful test for diagnosis of diabetes, and that this statement is false. Okay, so second popular belief I want to talk about, and this one was sort of directly related to this change in 2010, related to the recommendations for use of hemoglobin A1c for diagnosis of diabetes. And this is a controversial change. And one of the major sources of controversy, which is actually sort of a surprise to me, was that a lot of individuals, a lot of experts were concerned that hemoglobin A1c, and this was claimed in multiple statements, that hemoglobin A1c is a less accurate diagnostic test in African Americans. So I'm going to show you evidence today from our studies, and there's a number of other studies out there that suggest that hemoglobin A1c is an accurate test in both blacks and whites, and regardless of your race or ethnicity. So as I mentioned, so when hemoglobin A1c was first recommended for use of diagnosis of diabetes, this was actually very controversial. I mean, there's those of us who love hemoglobin A1c, thought this was very intuitive, we're not taking anything away, we're just adding hemoglobin A1c to the arsenal of tests that can be used. So I think I was a little bit surprised that this was met with such controversy. Turns out physicians, particularly older physicians, I would say, don't like change. A lot of people were very unhappy with this change, and people would come up to meetings and they were like, this is terrible. And I was like, I just do research. I'm an epidemiologist. I had this sort of like, I'm agnostic. And they were sort of like, this is all your fault. And I was like, you can use glucose. Nothing wrong with glucose. But so it was just interesting to me how controversial this was. And some of the controversy was related to the interferences in the assay. So there are some hemoglobin traits that can interfere with hemoglobin A1c assays. With the sort of quote-unquote common hemoglobin variants, HBC, HBS, with respect to modern hemoglobin A1c assays, not a problem. So major hemoglobin variants do not interfere with the modern hemoglobin A1c assays. There are still some very rare hemoglobin variants that are an issue. So yes, that's a concern. Actually, I'm told that most of the hemoglobin variants that we know, there's like 120 these days, right, have been discovered through A1c testing, and noticing that A1c test results don't jive with glucose levels. So that's definitely true, but I think a problem that can be managed and is not a huge issue. There's also conditions that can interfere with the hemoglobin A1c test, right? Absolutely. So anything that substantially alters red cell turnover can interfere with our interpretation of the hemoglobin A1c test. Right, but that's true with any biochemical test, right? Any biochemical test needs to be interpreted in the context of the individual patient and whatever other medical conditions may be present. And then also the expense, right? Hemoglobin A1c is much more expensive than glucose. So this is absolutely a concern. And in resource-poor areas, hemoglobin A1c may not be an appropriate or even feasible option for diagnosis of diabetes. And then finally, there was all of this controversy related to higher levels of hemoglobin A1c in African Americans. And the fact that there are higher levels of hemoglobin A1c in African Americans has now been repeatedly cited as a limitation of the hemoglobin A1c test for diagnosis of diabetes. And this was not sort of a scientific question that I set out to work on. It was kind of a debate I was pulled into by virtue of the fact that in the ERIC study, a quarter of our participants are African American. So we had one of the largest cohorts with a substantial amount of data on hemoglobin A1c in an African American population. And to give you some idea of what a number of experts were saying in the field, and this was getting a huge amount of traction. So these are some statements from the literature. The use of hemoglobin A1c for diagnosing diabetes or prediabetes in African Americans is ill-advised. Plasma glucose is more valid than hemoglobin A1c for diagnosing prediabetes or diabetes in Black persons. A1c may not be valid for assessing glycemic control across racial and ethnic groups. So very serious accusations. Okay, so on that note, you guys all know what Sesame Street is, right? So when I was a kid, I watched Sesame Street. And this whole controversy and debate reminded me of this segment. So there was this segment, and there was this song. I'm not going to sing the song, but it was a segment. They'd say, one of these things is not like the other. And there's this song, and then they'd show you 4 things. And you'd have to figure out which one of these things is not like the other. So here's an example. Okay, so let's play the game. Okay, so I have 4 things here, and I think one of these things is not like the other. Okay, so these are the cited disadvantages of hemoglobin A1c as a test for diagnosis of diabetes. So we've talked about interferences with the assay, some medical conditions that can interfere with our interpretation of the test results, its higher expense and low availability in some areas, and then the higher levels of hemoglobin A1c in African Americans. All right, so what do you think, right? I think it's the higher levels of hemoglobin A in African Americans, and the count, he agrees with me. Okay, so this controversy about how we interpret the higher levels of hemoglobin A1c in African Americans became such a sort of thing that not only did I sort of set out to publish a lot of papers and sort of really investigate it, but actually went around the country debating this topic. So in the American Diabetes Association in Boston and at the clinical chemistry meeting in Atlanta, Dr. Bill Herman and I went sort of face to face and debated this topic. And this sort of wasn't your usual scientific debate because we actually completely disagree. Bill does not agree with me at all about this topic. So this was quite the debate and very fun. And so my position in the debate, Bill's not here, so I don't have to give his position equal time, is that race does not alter hemoglobin A1c independently of hyperglycemia. That hemoglobin A1c is an equally good measure regardless of your race, ethnicity. And this, my side of the debate was informed by my own research and research of others, showing that yes, there are higher levels of hemoglobin A1c in African Americans compared to whites. There are a lot of racial differences in a lot of different biomarkers, and that higher level of hemoglobin A1c in African Americans compared to whites reflected risk. Right? That higher level meant that African Americans are at higher risk of diabetes and higher risk of complications, and those are things that we know. And so we set out to sort of formally compare the prognostic value of hemoglobin A1c in different racial and ethnic groups. And what we saw is that the association of hemoglobin A1c with retinopathy, with kidney disease, with cardiovascular disease, the associations, its prognostic value, its predictive value, was the same regardless of your race, ethnicity, suggesting that the higher hemoglobin A1c levels in racial or ethnic minorities suggest higher risk, higher hyperglycemia, and that we should be concerned. And that we should not withhold the use of this test in ethnic minority populations. All of those papers did not end the debate. And actually, I'll point out here, so I'm not in the habit of putting the conclusion of my paper in the title. This is the only 2 times I have ever done this. But again, this is this like null result that's really important, right? No ethnic differences in the association of glycated hemoglobin with retinopathy. So I made an exception for these 2 papers, trying to get this point across, that this very important null result, that it's an equally valid test in different populations. And actually, this work was done with Julie Bauer, who's now an assistant professor at Ohio State University, was a postdoctoral fellow at the time. Some other work that we did trying to get at this issue from another angle, this work was done with Christina Parinello, who was a PhD student at the time, now in New York. And so we looked at other measures of hyperglycemia, so serum markers of hyperglycemia. So these are hemoglobin-independent markers, not affected by hemoglobin, and we showed similar racial differences, suggesting there's nothing special about hemoglobin A1c or hemoglobin-based biomarkers. We saw the similar racial differences there. So to summarize, what we found is that hemoglobin A1c predicts microvascular outcomes and macrovascular outcomes similarly in black and white adults. We see black-white differences or black-white disparities in hemoglobin A1c, but we also see them for other serum or non-hemoglobin-based glycemic markers. And so that suggests that the disparity may not be specific to hemoglobin A1c and may be primarily hyperglycemia. Maybe what we're seeing is a glycemic process, and that the higher hemoglobin A1c values in blacks likely reflects higher risk of diabetes and higher risks of complications. And that supports a similar interpretation, similar cut points, similar use of hemoglobin A1c in blacks and whites for diagnosis and treatment of diabetes. Sort of despite these publications, these debates, me making a lot of noise about this, it's sort of been interesting that this is still, this idea that race, ethnicity is a limitation of the hemoglobin A1c test has really gotten a lot of traction. It's made its way into various statements and guidelines. And it's just very concerning to me that we might withhold or use a test differently based on race or ethnicity. And I sort of pondered this because I think some of this may be a communications issue. And also that humans, I think as humans, we see differences, and we're just kind of programmed to see differences in patterns, and then we attribute meaning to those differences. And so I've been going around, with this kind of null result, this boring message that yes, there's a difference, but it doesn't actually mean anything important, right? So this was a perspective I published a few months ago where I sort of summarized the whole controversy and debate, and I said a difference to be a difference must make a difference, which is a quote attributed to Gertrude Stein. And it's sort of interesting to me because for some reason this just seems too subtle for many people. And so this what I think is a misconception has gained a fair amount of traction, despite, I think, all the evidence would suggest that hemoglobin A1c is not a less valid test in African Americans or other racial or ethnic minority populations. Okay, so the third misconception I want to talk about is one that you may have heard about. I don't know if you've seen headlines or other news reports or press releases that have suggested that over 1/3 of diabetes in the United States is undiagnosed, right? And this suggests there's a lot of people out there who are walking around with undiagnosed diabetes. And so this is a very common statement that you see. I'm going to try to demonstrate today some evidence that suggests that actually this is not true, that these conventional estimates that we think about for undiagnosed diabetes are actually inaccurate, and there's only a small proportion of the population of people who are undiagnosed. And that's good news, right? It means we're doing a good job with diagnosis of diabetes. Okay, so let me walk you through some of this evidence. All right, so I showed you the 2010, that was major change to clinical practice guidelines and how A1c had been added to our guidelines. So this is the 2016, whoa, why is that happening?
How do you do that?
I don't know, that's interesting. Okay, are you dizzy? All right, maybe I won't touch anything. I know I'm not allowed to use the pointer thing, so. Okay, so I'll point. All right, so in 2010, hemoglobin A1c was added for diagnosis of diabetes. We've been now using hemoglobin A1c for the diagnosis of diabetes for the last 6 years. This is the 2016 report, clinical practice guidelines, and you can see here the criteria for diagnosis of diabetes. You'll notice that things are no longer numbered, okay, and that hemoglobin A1c has been demoted, right? It's now at the bottom instead of the top. Try not to take that personally. It may have something to do with Mr. Bill Herman, Dr. Bill Herman being on the committee, I'm not sure. And the lack of numbering is designed to reflect the idea that no one test is preferred over the other. But I want to point out here is that this footnote at the bottom, okay? So at the bottom in these clinical practice recommendations, it says results should be confirmed by repeat testing, okay? So in our clinical practice guidelines for diagnosis of diabetes, the ADA is very clear that unless there's a clear clinical diagnosis, a second test is required for confirmation. All right, and they also state that if two different tests, such as hemoglobin A1c and fasting glucose, are both above diagnostic thresholds, that confirms the diagnosis of diabetes. Okay, so the ADA explicitly recommends confirming a diagnosis of diabetes. So let's talk about, so why am I so concerned about this idea of confirmation? Well, that's because, as I've mentioned, there's high variability in 2-hour glucose and fasting glucose, and especially relative to hemoglobin A1c. So if you use one test, if you use a single fasting glucose to identify elevated glucose, elevated hyperglycemia to make a diagnosis of diabetes, you will capture a lot of false positives. All right? And so guidelines for diagnosis of diabetes recommend 2 measurements to confirm the diagnosis to eliminate or to reduce the probability that we're capturing false positive cases. So if only a single measurement is used to define diabetes, prevalence estimates will be inflated because you'll have all these false positive cases in there. So this confirmatory process, this process of using multiple tests, is designed to get rid of those false positives, and it means prevalence will actually be lower if we do that. So conventional definitions in prior epidemiologic studies have not attempted to confirm undiagnosed cases of diabetes. So that means that previous estimates of prevalence are substantially overestimated. And this has major implications for understanding the proportion of total diabetes cases that are undiagnosed. And I think that's why I'm sort of focused on this, is because understanding the proportion of cases that are undiagnosed out of total diabetes cases is really important to understand how we're doing with screening and diagnosing of diabetes in this country. So some solutions to dealing with this variability, this inherent variability in our glucose tests, is to repeat measurements as recommended by the ADA, and to use definitions in our epidemiologic studies that closely approximate those that are used in clinical practice. So these are some data from the National Health and Nutrition Examination Survey showing you the increasing prevalence of diabetes in the United States. And what I want to point out to you, so this is total cases of diabetes, and then this is using a definition of undiagnosed diabetes that is very consistent with current clinical practice guidelines, where we have confirmed elevations we've confirmed hyperglycemia in these individuals. And so this black box down here is the proportion, this is the prevalence of undiagnosed diabetes. And so one of the things to note is that as a proportion of the total, the proportion of undiagnosed cases has actually been going down. And I'm going to show you this data on the next slide, showing you the most recent data. So this is the percentage of total diabetes cases that are undiagnosed. So these are the same data as on the previous slide. And so what we see is among the total cases of diabetes in the United States, only about 11% are unrecognized or undiagnosed. Okay? So this is actually really good news. This suggests we're doing a very good job with screening and diagnosis of diabetes. That if you use a definition that is similar to what we would use in clinical practice, then very few people are walking around the United States with undiagnosed diabetes. But this isn't how anyone else has estimated the prevalence of undiagnosed diabetes. So our conventional estimates used by Centers for Disease Control and NIH and other organizations have published estimates where over 30% of cases of diabetes are undiagnosed. And I think this is a serious concern, because this is a totally different message, right? This suggests we are not doing a good job with screening and diagnosis, and that there's a substantial proportion of the population walking around with undiagnosed diabetes. So some implications of this, right? So what we see is that conventional definitions in epidemiologic studies haven't conformed to clinical practice, and I think this is a problem. There's actually a simple solution to this. We can use definitions that more closely approximate clinical practice. We can use definitions to confirm cases of diabetes in epidemiologic studies. And so if we use accurate definitions, this demonstrates that we're doing an excellent job of screening and diagnosing diabetes in the US, which I actually think is a very positive message and suggests we're doing a great job in clinical practice. So are over 1/3 of diabetes cases in the United States undiagnosed? Looking at the same data and using more accurate definitions suggests that's actually not the case at all. Only a small fraction of total diabetes cases are undiagnosed. Okay, finally, I want to talk a little bit about prediabetes. So a number of people out there think prediabetes is not real. And a lot of folks have said it's just a condition created by the pharmaceutical companies to make more people eligible for diabetes drugs. So I'm going to talk today about prediabetes. Hopefully I can demonstrate to you that I think it's real, it's associated with substantial risk, and that lifestyle interventions can improve outcomes. So some quotes from some of my colleagues stating that prediabetes is not a medical condition. My friend John Yudkin really does not like the term prediabetes. He thinks it's an artificial category with virtually zero clinical relevance. And actually, when John writes anything about prediabetes and submits it to journals, he always puts quotes around prediabetes, and then the journals remove all the quotes. And John really does not like the term prediabetes. He and I haven't actually gone around and debated this topic, but we have a lot of fun discussing it. So what is prediabetes? So we've discussed that hyperglycemia and its risks exist along a continuum, right? And so prediabetes is a term that we use for individuals who don't meet the threshold for a diagnosis of diabetes, but are just below that threshold, right? So these are people who are very high risk for conversion to diabetes, but don't quite meet the criteria for a diagnosis of diabetes. We've shown in prior studies that there have been major increases in prediabetes over the past couple of decades. But one of the big difficulties in the field right now is there are presently 5 definitions of prediabetes that are in current use, okay? So 5 definitions of prediabetes based on glucose, 2-hour glucose, or hemoglobin A1c using different cut points. So currently, the WHO, ADA, other international organizations, no one agrees on a definition of prediabetes. So this is a major difficulty in the field and causes confusion in clinical practice, and also makes, Bethany Warren, who published this elegant paper just a few weeks ago, can tell you it also makes papers very hard to write because you have 5 different exposure categories for every analysis that you do. So this was a paper that led by Bethany Warren, who's a PhD student here. I really love this paper. It's a very elegant paper where she looked at the different definitions of prediabetes. So all different 5 definitions, and looked at what these definitions mean for future prognosis in these individuals using data from the Atherosclerosis Risk and Community Study. So these are some of Bethany's data. So just showing first the prevalence of prediabetes. So you can see prediabetes is very common in this middle-aged population, but how common really depends on which definition you decide to use. So it varies widely depending on the definition. This is the relative risk of incident diabetes, future diabetes, chronic kidney disease, cardiovascular disease, and all-cause mortality among people with prediabetes, looking at these different definitions. And so what you can see here is that regardless of the definition, these individuals are high risk, very high risk for the subsequent development of diabetes. Generally, across all definitions, folks with prediabetes are also at high risk for chronic kidney disease, cardiovascular disease, and all-cause mortality. So we see, high morbidity and mortality among people with prediabetes. So I think, is prediabetes real? I think the answer to this question is yes, prediabetes is real. It's a high-risk state. It's associated with substantial morbidity and mortality. I think a really important message is that prediabetes provides an opportunity. a lot of our interventions work much better early in the disease process. So the earlier you are in the disease process, the more effective our interventions are. So I think prediabetes can provide an opportunity to prevent diabetes, intervene to address lifestyle factors, address cardiovascular risk. We know from randomized clinical trials that even modest weight loss can reduce the subsequent development of diabetes. Prevention of weight gain, also a very important goal. So I think, in summary, prediabetes provides a very important opportunity, and we shouldn't ignore this condition. However, I think having 5 different definitions and no agreement and a substantial sort of disagreement across international organizations regarding optimal definitions is a problem for the field. And then I think a next step is to think about how to move forward towards consensus and coming to agreement in how to define prediabetes to help move this field forward. So prediabetes, I think, is a real condition and that is not a useless term. Some advice for all of us that I think that we should keep in mind. I think in general, I think evidence-based policy is very important. And I think epidemiologic evidence is really critical to inform both public health and clinical policy. And I've really tried to spend my career doing the type of research that will directly influence clinical practice and clinical guidelines, how we diagnose diabetes, how we identify people who are at risk in the population. So some conclusions, some facts from today. So hemoglobin A1c, for those of you who have not been to one of my lectures before, now you know, is a helpful diagnostic test, and I think it provides important and prognostic information. The overwhelming evidence suggests that as a test, as a diagnostic test, and as a measure of glycemic control, hemoglobin A1c does an equally good job, regardless of your race or ethnicity. The current evidence from national data suggests we're actually doing a good job with screening and diagnosis of diabetes. The proportion of total cases of diabetes that are undiagnosed in the United States is relatively small. I think prediabetes is a growing problem and really should be a target of our efforts to prevent diabetes. I don't know if Naresh is here today. He showed up last week, actually, for my lecture, so I'll give him credit for that. But he and I are involved in developing a randomized clinical trial, and I think for a novel intervention to prevent progression from prediabetes to diabetes. And I think this is where we need to be focusing our resources and efforts, is thinking about how do we prevent diabetes in the first place. But I do think if we're going to really spend a lot of time tackling prediabetes, it would be good to come to some global consensus on how do we define the condition. So I have many, many people to acknowledge, way too many to list here. I do want to mention Fred Brancati, who passed away in 2013, who was my very close mentor and very important in my career. Joe Koresh, who's here today, who first recruited me to Johns Hopkins as a PhD student. Mike Steffes has been my close laboratory collaborator at the University of Minnesota and is as a hemoglobin A1c guru. The Welch Center has been my academic family, and I really want to thank all the staff and faculty and trainees at the Welch Center. And I think it's very important to thank, I really appreciate all the contributions of staff and participants of all of our different studies, because none of this would be possible without them. Especially want to thank Alex and Bethany, who've been a major part of all this research for the last couple years. My 2 most important projects I actually didn't mention. They know who they are. Ben is 8 and Eli is 5. And epidemiology is a team science, and I have an absolutely fantastic team. So thank you all for coming today and for listening to this lecture.
Liz, that was great. So I just want to ask everyone to use a microphone at either side, because this is being recorded and we want to make sure the questions are on the podcast. So let me ask a couple questions. You referred several times to modern modern test for hemoglobin A1c. So the classic test, electrophoresis, you look at mobility, and if there's a lot of glucose in the hemoglobin, it doesn't move as fast. How are modern tests different than that?
I'm not a clinical chemist, so I'm not going to give you a very.
But it's electrophoretic?
But it's all HPLC for the most part.
I
There are
see.
some, so immunoassays are not really used anymore.
I see,
But
okay.
almost all HPLC assays,
Okay.
yeah.
And then, you said several times, you showed that slide that hemoglobin A1c measurement is one test. You didn't say it was a better test for a diagnosis, but if it's more stable, less variable, there should be less misclassification, right? And it should be a better test.
Yeah, I mean, I think I'm trying to be diplomatic here.
but have you looked at the-
I think my bias about hemoglobin A1c is well known. I think.
But have you looked at, test characteristics, area under the curve?
Oh, sure. So, I
ROCs?
mean, one of the things is, to be fair to glucose, right, is that when we only have one value, right, if we only have one hemoglobin A1c and we only have one glucose in an individual, hemoglobin A1c will always win.
Right.
Because of this, because it's more stable and because it's more reliable. So from an epidemiologic standpoint, right, it's fantastic.
Right.
It's this integrated measure and, successfully ranks people in the population and is associated with risk and is a very helpful biomarker. On the other hand, it's an indirect measure, right? So, and there are things, non-glycemic factors that can affect hemoglobin A1c. So, glucose is a direct measure of what is diabetes. So I think that is also important to recognize. It is true, if you have multiple glucose tests and you put them head-to-head with A1c, things really do start to converge and you don't see much difference between the two.
I would say it is indirect, but it's an integrative measure.
It is an integrated measure, yeah.
So I think Al Sommer here also wants to ask a question, so I'll encourage people to stand up.
Other people other than the deans can ask questions.
So that was absolutely terrific, Liz. I just wish you'd show more enthusiasm during your talks. You're so shy and retiring, So let me ask you a basic question. One which, since you started with diabetic retinopathy, that
How did I
brings us
guess
to
you
the
would
eye,
ask
of
about the
course.
eye?
But I'm not going to do retinopathy. I'm going to do glaucoma, which
Okay.
is my favorite eye disease. And there we've had this big thing of low-tension glaucoma, pre-glaucoma, what have you, based upon intraocular pressure. And what I've spent most of my life uselessly doing is trying to convince people that this whole thing is a gradient, that there's hardly ever a biologic measurement that's a dichotomous. So when you talk about pre-diabetes, have you ever looked, you have to have looked at if the hemoglobin A1c is 4, 4.5, 5, 5.5, 6, 6.5, and then for each one of those, what is the risk? I would guess, but you'll prove me wrong, that it's in fact a continuous variable, and then you're just making a relatively arbitrary cost-benefit ratio that we're going to cut it here because the risk is X and you're going to have so many false positives, false negatives. But I'd like to hear what you have to say
Yeah,
about that.
so absolutely. So that's a fantastic comment and and a great analogy. So absolutely, risk for glucose, for hemoglobin A1c is along a continuum. And so there is this inherent sort of paradox between kind of drawing this arbitrary cut point and defining people above and below that threshold as having a disease versus not, or having a condition versus not, when everything is along a continuum. So there is, and biostatisticians who don't have to deal with things like diagnosis and some of the practical realities will often say, this is a terrible idea. Do not put your data in bins. There's tremendous loss of statistical efficiency. It's a terrible idea. On the other hand, right, in clinical practice, the diagnosis is absolutely necessary. So putting people in that box is sort of fundamental to clinical practice, even if it is somewhat arbitrary, because without a diagnosis, you can't have reimbursement, you can't have treatment decisions, you can't have referrals. So that diagnosis is sort of necessary for clinical care. And so we draw these arbitrary thresholds and we say, okay, these people have diabetes, even though we know that someone with a hemoglobin A1c of 6.5, is not that different than someone with a hemoglobin A1c of 6.4, right? And that risk is along a continuum. So absolutely, I think it's necessary, but it's sort of a necessary evil in a sense. For epidemiologically, the risk is along a continuum, and I can analyze things along a continuum and look at all the distributions. But in clinical practice, we have to say this person has diabetes, this person has prediabetes. And prediabetes is sort of an example because everybody's come up with different definitions because it's along a gradient, and there isn't a magic number, and the data don't tell us that this is the number we should use. So therefore, there's tremendous confusion. You don't know, well, should this person be referred to nutritional counseling? Will this insurance company reimburse for weight loss counseling or other types of intervention? So that lack of having a clear box actually creates a problem in clinical medicine, although not a problem for us epidemiologists because we're happy to analyze things along a continuum.
Great. Well, I'm going to ask another question. So could you go back to...
Other people other than the deans can ask questions.
Could you, well, they're all your friends. They all talk to you about this all the time. I think. Go back to the Gertrude Stein difference.
The difference to be a difference must make a difference?
Yeah.
I don't know. There we go. Here we go.
So these racial differences in HB, that last sentence, have not been explained by differences in glycemia.
Yeah.
If they really represent, an increased exposure in African Americans, when you get multiple measures of glucose and adjust for it, the hemoglobin A1c level should be the same, right? But that's not what that says.
Yeah, so this is the problem. So we don't have large studies with African Americans where we have multiple glucose measures. So that's a fundamental problem. So what people have done have looked at hemoglobin A1c in blacks and whites, and then they adjust for fasting glucose. And when you do that, the difference doesn't actually go away. It diminishes, but it doesn't go away. And so that's been used as this argument. Well, look, this difference is independent of hyperglycemia. But going back to sort of your original question, we know that a single fasting glucose doesn't represent true average glucose in an individual. And so it may actually be that certain racial ethnic minority populations have higher postprandial glucose that's not at all reflected in that fasting glucose, but is reflected in the hemoglobin A1c. And so that's why we see these sort of bigger disparities with hemoglobin A1c. We also see them with some things like fructosamine, glycoalbumin, which are other integrated measures of average hyperglycemia. We don't see much of a difference with fasting glucose.
Go ahead. Wait, you got to use some more. Al, you
so
can't
now I understand what a dean's lecture is.
To go back to the glaucoma analogy, in fact, when we look at the prevalence of glaucoma in whites and African Americans have a much higher level of glaucoma, and they get it earlier in life. But if you look at the distribution of intraocular pressure levels of those who do not have diagnostically proven glaucoma, they're identical. So, it sort of works with your your glycemic level and your hemoglobin A1c, that when you have the disease is there, and you will have the underlying physiologic determinants, but when you look at the population as a whole, there's no difference between the population.
Okay. Peter. Yeah, Peter.
So a non-dean question. First of all, that was a terrific lecture. Thank you. Just the basic science here. Who discovered A1c, and when did they first put it together in terms of a tool for managing diabetes? And is it equivalently useful in juvenile and type 2 diabetes?
So great question. So David Nathan, I don't know that he was the discoverer of hemoglobin A1c, but he was sort of the main person who brought it to clinical use and did some of the seminal studies in the late 1970s and early 80s. Chris Sodak was also involved, who was an endocrinologist here, who passed away a few years ago. Really brought it to show that it was a measure of hyperglycemia and could be a useful measure in the setting of diabetes. So the second part of your question?
Management of type 1.
Oh, management of type 1. So we have, so A1c is a standard measure used to monitor type 1 as well. We don't have as much data in kids. So for diagnosis, all of the guidelines are kind of wishy-washy. Because we don't have sort of the type of data like we have from the ERIC study, where we have sort of an initial population and then who are non-diabetic and then followed for a long time to see what happens. So we don't have those type of data in kids to show sort of what different levels of hemoglobin A1c mean for future prognosis. So right now, at least for type 2 diabetes in kids, we generally say we think the same levels are probably reasonable, but the evidence base simply is not as strong as for adults.
Thank you. Next question, please.
Thank you for the great lecture. My question is talking of the idea that this measurement, like that Dan mentioned earlier, is on a gradient level. And we do know there are some markers that if you see a high elevation of these markers in certain populations, it's totally normal. It's not a cause for alarm. Whereas in another population, you get the same amount of gradient of that marker and you need to worry. So my question is it possible that maybe in the racial differences or gender differences or maybe geographic differences, is it possible that maybe hemoglobin A1c may have this particular measure, let's say 6.5 in a population, but it doesn't cause any problems, and in other populations, that same level is raising problems?
Yeah, so that's a great question, and that's really sort of central to this debate, and so that's why we've sort of been going around to different cohorts to try to answer that exact question, is does that same level of hemoglobin A1c mean the same thing for future risk in these different populations? And thus far, what we've shown is that yes, a hemoglobin A1 6.5, a hemoglobin A1c of 7, has the same amount of risk associated with it regardless of the study population, regardless of the racial ethnic makeup of the study population, and that it looks like it's a similar prognostic test. And this is really what's been central to this discussion, is whether we think the difference or the elevation in hemoglobin A1c in certain racial and ethnic minority populations is artifactual, right, or whether it's hyperglycemia. And I think it's hyperglycemia, and at least the data that we have and the studies that we published and a number of other studies in Japan and other populations have now shown that those racial differences actually mean higher risk. And so at least at this point, on the basis of current evidence, I think it suggests we should use the same cut points, right? Some investigators, some experts have advocated different diagnostic cut points by race. And so I think that at this point, there isn't evidence for that we should use it similarly. And I also think that for me, there may be some non-glycemic determinants of hemoglobin A1c that are important, right? Red cell turnover is important. We know that red cell turnover can affect hemoglobin, and as to the extent that human beings differ in their red cell turnover.
The red cells, the hemoglobin doesn't hang around long enough to be exposed to the glucose.
Exactly. So I think to the extent that there is variability in red cell turnover, that could be important. Unfortunately, we cannot measure red cell turnover in population-based studies. It's very, very hard to measure, even in small, in one individual. So we don't have measures of red cell turnover. So I think if we were to try to correct A1c and maybe some of the differences that we see, being able to quantify red cell turnover would be fantastic. We don't know anything about differences in red cell turnover across populations. So to me, using race as a surrogate for for red cell turnover or some other non-glycemic factor is actually very disquieting. I think that we should be moving away from race-based medicine. We should not be saying things like, A1c is less valid in certain racial ethnic populations. I think that if there are important non-glycemic determinants of hemoglobin A1c, we should figure that out, and we should try to use that to improve its use. But I think using race as a surrogate for some unknown biological factor is a really bad idea.
Okay, so let's go with the last question, Josh. Because we have a reception afterwards. I want to make sure everybody has time.
Well, that's why they're here, right? For the food.
That's why they're here.
Well, hypoglycemics, yes. So that was such a great lecture. I was struck by the fact that as your data seemed to get stronger, the policy side seemed to get weaker.
Well, that's just because it's newer. No one, they haven't come to me yet to sit on the committee to make the new policies.
Well, that's actually my question. I mean, you think like as the data gets stronger, the policy gets weaker.
Yep.
Maybe that'd be par for the course for Congress, say, right? But for the world of epidemiology and data, you don't expect to see that. What do you think is it just the personalities? How do you make sense of the fact that you're proving in cohort after cohort and you've got study after study, and yet suddenly, hemoglobin A1c is number 3 on the list, and how what do you think is going on?
Well, it's still on the list. So, and actually, just and I think it's being widely used. I think most people and the senior at the senior levels of the ADA and other major organizations, I think, are very much agree with a lot of the statements I've made here today. It's interesting. Translation is this key word now. It's actually interesting to me that the type of translation I do, the science gets very, very boring in order for it to be translated. We have to do many, many studies. The novel study is not what gets into clinical guidelines, right? It's that 10th great big study that, as Mark Kalushko always says, don't we already know this, right? I think that's what he said about the New England Journal paper, right? So it's like once we show beyond the shadow of a doubt, that's when the translation happens. So I think it is somewhat related to the uncertainty in the field. This issue of the racial differences thing I think has a little bit to do with race. Race, like sex, people seem to have a different attitude towards controversies in those areas. They tend not to get tamped down quite so quickly as more boring things like diagnostic thresholds. So I think, and it's interesting to me, I think it's also just a lot of politics, right? I think that the folks who are making these claims about racial differences don't do a lot of big epidemiologic research or not doing a lot of the evidence, but they're generating a lot of noise and they've gotten a lot of traction. And I don't know that, and so, I'm kind of boring, right? I just sit in front of my computer and I do epidemiology. So it's somewhat hard to compete with these sort of big booming physician voices out there claiming there's a difference, this is important, and me saying no, there's a difference, but it's not, it doesn't really make that much a difference, and look at all these null studies, and it's sort of not sexy. So I think that message has been drowned out. But I'm still trying very hard.
Well, I think it's sexy. Being an epidemiologist, I love data. I thought it was a great talk, and I'm sure there's going to be. I have lots more questions for you,
Okay.
But I want to. It was just a great presentation,
Thank you.
And please join me in thanking Liz and congratulating her.
Thank you very much.